Hey everyone, welcome back to our next lecture over DNA technology. So this lecture is going to address how science manipulates DNA, some of the ways that science uses DNA to help solve crimes and things like that, and some of the ways that science can manipulate DNA to alter species and genetically modify organisms. So let's go ahead and get started with that. So DNA technology is super useful in all kinds of fields throughout the planet. Every single thing that you do on a daily basis, from the things that you eat to the clothes that you wear, and the antibiotics and things that you take, are probably going to be influenced by some sort of modern day DNA manipulation or of some sort, which we'll talk about in a little while. Research, medicine, agriculture, criminal justice, and all kinds of stuff uses DNA within those fields. So you've probably come into contact with something today, at least multiple times today, more than likely, that has been altered or had its DNA used for something while it was being made or for its purpose. So scientists can manipulate DNA. We know how it's put together. As we've talked about in previous lectures, we know what the structure is. We know how to alter the structure. We know how to read the code. We can alter the code as well. So being able to alter that code changes the outcome of the protein that's produced from this DNA sequence here. Once the DNA sequence is changed, the protein is different, which means it does something different. So sometimes we can change that DNA to make a protein that does something totally different and something different that we want it to do. So it might not be able to do something from the get-go, but when we change it now, it can do something different. And that's the outcome that we're looking for. So manipulating DNA for practical purposes is called DNA technology, and that's what this lecture is going to cover. So if we read this DNA, which scientists can do very easily, every single organism on the planet has tons of DNA inside of it. And we can read the unique sequences of those DNA, which will allow us to figure out the evolutionary history of organisms, or if they have a particular disease, we know what the normal gene looks like, and if it's an altered gene, we know it results in this particular type of disease, and things like that. So very, very, very useful for also helping figure out who's related to who, family-wise or DNA-wise, genetically, and things like that, evolutionarily. Very, very useful. Help solve crimes, diagnose all kinds of medical conditions by DNA. So very, very useful studying the DNA. So lots and lots of ways that scientists and biologists in particular are going to manipulate DNA. Tiny little pieces of glass tubes, entire genomes can be altered at once, lots of different ways. So one of the ways that every single species on the planet uses the same genetic code, the A, C, T's, and G's thing. You're probably familiar with that one from our previous lecture. And traditional organisms have just the genes from their own species inside of it. So this fish would generally have genes from its mom fish and its dad fish that are both the same species. Well, if you take the genes from an organism that doesn't have, or that's not related to this one, so this fish has jellyfish genes inside of it, which make it glow. So those jellyfish genes have been inserted inside of it. Those jellyfish genes are obviously a different species than this original fish. So now this fish has genes from two different species inside of it, the original fish that it is, as well as that jellyfish. And since every single organism on the planet is made up of the same A, T, G's, and C's for their DNA, it's really easy to splice in different genes and things because essentially the words are the same. If two people speak English, it's essentially this DNA is written in English, the jellyfish genes or DNA is written in English. It's pretty easy to splice words together to make sense. So it's pretty easy to stick DNA from one organism inside an organism that's some sort of closely related to it. It doesn't have to be super closely related to it. Jellyfish and fish are kind of closely related to one another, but not super. So it really just depends on the organisms in question, but super cool that you can do this. So when organisms are combined with more than one species, they tend to be called recombinant DNA. So bacteria are a way that transgenic bacteria that have more than one species of DNA or more than one original gene inside of them, are super useful. So we can take genes from one particular species of bacteria that has the ability to break down petroleum, for example. But the original bacteria that breaks down petroleum is super dangerous to humans, or maybe kills fish. So what we can do is we can take the gene out of this particular bacteria that eats petroleum. So we can take the gene for petroleum degradation out and stick it in a very neutral bacteria, one that doesn't harm fish. So if there's an oil spill in the ocean, we can pour those engineered bacteria into the ocean. They'll break down the petroleum, get rid of the oil spill, and not harm the fish in the ocean, which is super cool. So very, very, very useful. Drugs like insulin is produced this way. We take the gene for human insulin from the human's pancreas cells, the gene to produce insulin, and we splice it in E. coli, which is a bacteria. So then that E. coli now starts producing insulin instead of what it's normally should be producing, which is super cool. Lots of other genes are produced inside of bacteria, lots of other medical products. Things like blood clotting factors, warfarin, heparin, the blood thinners, fertility hormones, and things like that. You can also make bacteria, like I said, that can break down petroleum, ones that can break down pesticides, and other natural things that might occur in the soil. So very, very, very useful. Bacteria are very easy to manipulate. They're usually pretty simple DNA speaking wise anyway. So they're pretty easy to manipulate. They grow really quickly, so they're really good things to manipulate their DNA with, to play around with. So how do we do this in organisms? How do we make these strange organisms? These transgenic organisms, ones that have different DNA inside of them. Well, this is called a plasmid. Now, plasmids are little tiny pieces of DNA that kind of work like an extra to a chromosome. They're not really chromosomes, but they're kind of just like add-ons to chromosomes, and lots of species have them. But since it's DNA, if it gets inside of a cell, the cell will use this DNA to make whatever the plasmid has. So the little plasmid sometimes contains genes on it, and the genes can be for making antibiotic resistance if you're a bacteria, or producing insulin like that if you're a recombinant, things like that. So we take the plasmids, these things occur naturally, and then we put genes inside of the plasmid. So we insert whatever gene we want. So bacteria don't originally produce insulin, obviously. They don't need it, they don't use it, they don't make it. So what we did was we took a plasmid, we cut it in half in a lab, and we took the gene from humans that's going to be the gene that causes humans to produce insulin. We cut that out of the human DNA, and we stuck it inside of the plasmid. Now this plasmid is essentially the letter that allows us to get it inside of our bacteria. This is the delivery vehicle. So without the delivery vehicle, essentially a letter just shows up and nobody reads it. You got to have the mailbox, or excuse me, the envelope, the delivery method to get it there. So the letter, what we want inside, has to be put inside of the envelope of the plasmid to get to the bacteria in the first place. So we cut out the gene we want from the human gene, stick it inside of a plasmid, and then we give it to a bacteria. So that bacteria then gets the plasmid inside of them. They take that plasmid, and then they start reproducing themselves with that plasmid inside of them. More and more little babies are going to be produced with the plasmids inside of them. And now the bacteria is going to have a plasmid inside of it that has the gene for producing human insulin inside of it. So now that that gene for human insulin is inside of the bacteria, it will now start producing human insulin. So we stuck a gene from human inside of a bacteria. This bacteria will now produce human insulin, which we can purify away and use to help people that have diabetes. We used to have to harvest this from dogs or pig livers, and that was very, very medically expensive, not particularly ethical, and not particularly pleasant to do either. So this is significantly easier, cheaper, and way faster. So bacteria are very, very, very useful in this ability to put genes inside of a different species from one species into another is also very useful as well. There are lots of ways to move plasmids around. You can zap a cell with electricity, which causes them to uptake the DNA. You can use a tiny little needle called a gene gun that shoots the DNA directly inside. You can make little tiny bubbles, little cell membrane bubbles, essentially, that will be phagocytized and merge with the cell membrane and essentially deposit the plasmid inside of the cell for you. Or if you recall from our previous lectures over transduction, a virus can infect a recipient cell. So very interesting ways to get the DNA and the plasmids inside of bacteria. They just don't naturally eat the plasmids kind of thing. You have to get them inside. So you can also do this with plants. You can make plasmids in a lab that contain herbicides, which are cool. Or you can have them that produce their own pesticides, or ones that are resistant to drought, or ones that are resistant to bugs and pests, which is super cool. So the bacteria introduces the plasmid to the essentially you put the plasmid inside of a bacteria and then you infect a plant. Once the plant becomes infected with the bacteria, the plasmid is essentially going to enter into the bacteria or the plant cell, and then the plant will now start using that plasmid. So the plant, once it reproduces, every single one of its offspring will have the gene as well. So all of the plants, the tobacco plant you can see here is the one that they're talking about, that they're using for example. The plasmid contains the gene for a herbicide. So now every single time that this tobacco plant reproduces, every single one of its offspring will be resistant to that particular herbicide, which is cool. So you can introduce herbicide resistance with a plasmid into plants or an animal like this, and then every time that that organism reproduces, its cells will make a copy of that original gene because the genes are in the DNA to start with from that plasmid. So the plasmid incorporates in the DNA, which is super neat. So it's like a permanent change. So if you see the glowfish at the store, they're not dyed; they are naturally that color due to this process. They put a plasmid inside of their cells, and that plasmid was uptaken by the cells. And then as the fish reproduced, the plasmid reproduced inside of their cells as well, and their babies came out naturally pink. So the original cells were altered, and then from there on out, all the rest of them stayed pink and glowing green and all those different cool colors. So very interesting how this works. You can use viruses to infect cells to give them DNA. So essentially, if you recall from our little lecture earlier about how viruses work with the transduction concept, essentially a virus is a postage envelope that carries a message inside of it. It does not matter what the message is, the virus will deliver it regardless. So if you take out the dangerous stuff on the inside of a virus and you put something good inside, like a gene to produce insulin or a gene to fight cancer or something like that, you can introduce that virus to the human or whatever animal it is, and instead of killing the human or the animal, now this virus doesn't have anything dangerous inside of it, and it just delivers a modified gene that's super useful. So it might deliver the gene, it might infect the host cell with the gene for resistance to a particular type of cancer, or the gene might be inserted that causes the host cell to change the cell membrane shape or something like that. So super cool what you can do with these. So you take the dangerous part of a virus out, put something good inside, and then you have a modified virus which can be then used to do good things instead of bad things, which is super cool. And then once again, since the cell is permanently infected, every single time that the new cell divides, the baby cells will now have that extra altered DNA inside. So this makes that permanent change again, the virus concept here. First off, to know how to read the DNA, you have to sequence it. So DNA sequencing is really cool. Essentially what they do is they take DNA and they run it through a big giant machine, and it tags the G's, T's, A's, and C's with different electrically sensitive dyes and things like that that fluoresce under different colors. And then as the sequencer runs, it essentially just counts the numbers of yellow and blue and green and purples that flash and what order they come in, and that's how they know what order the DNA is in. Super neat. So you can either do the entire genome, you can do tiny little pieces of this DNA, entire chromosomes, little tiny sequences of genes and things like that. But once you know what the letters read, then you can know what the sequence is, and you can know what the protein is being produced, how closely things are related, if the DNA is the exact same, or how far apart things are related, and things like that. So knowing what the code of the DNA is allows you to alter it, and it also allows you to tell who's related to who, and things like that. It's very useful. There's lots of different ways to sequence DNA. So you can see down here the cost of sequencing a human DNA in 2001 was about $100 million when they did the Human Genome Project. Today it's about $1,000 to sequence, probably a little cheaper now since this is a little older. So it's pretty easy. We can do this in our lab very, very easy. This is a concept called PCR, super easy, and then this is the sequencer machine. So pretty neat way to do this. It's a lot cheaper to sequence DNA than it used to be, which makes studying evolutionary relationships very, very useful and very easy at this point in time. And also it allows people to sequence DNA and study diseases and things like that to figure out what the genetic cause of that disease is and if we can fix it or prevent it in the future. So PCR is a super neat way to get DNA copies. So essentially what happens is you come into a crime scene and there's a tiny little speck of blood on the floor. That's the only thing that's left behind. You as the crime scene investigator collect that. There's a little bit of DNA inside of it, but you don't have enough to do a bunch of tests to figure out who the criminal is. And if you screw up your test, you're going to mess up the blood and then you're out and you're done. So PCR is a super, super cool way to make copies of DNA sequences. You can amplify a sequence and make millions of copies of DNA. So you can make that one copy or 2 or 3 copies of DNA that you found in that blood droplet into a couple billion pretty easily, which would allow you to have way more to test with. And if you messed up or if you had an inconclusive result or something like that, that would allow you to have extra to deal with, which is a very cool thing. So PCR is super neat. You have a target sequence, promoter regions, and things like that. I'm going to rely a little bit on the supplementary videos here to so you guys can see this being walked through how this actually works. It's super neat. You take one copy, essentially little things called primers come in, they bind to a specific sequence. DNA polymerase comes in, finishes out the chain, so now you got 2 pieces, and then 2 to 4, 4 to 8, and then 8 to 16, and so on and so forth. So every time that this goes on, you get double the amount of DNA that you had to start with. So very, very, very useful process, PCR. Like I mentioned, it copies DNA over and over again, and you can make millions of copies of DNA very, very quickly, very cheaply. It takes about 4 or 5 hours to make billions of copies and can be completely done because you have to heat the machine up and cool the machine down. So that takes a little while. So super neat. You can use this also for detecting, like I said, tiny little pieces of samples and things like that. You can amplify it. It's also useful for positive or negatives sequences. So if you are looking in a stream for a particular species of fish and you're just looking for that DNA, if you know that there's a particular DNA sequence that's unique to that fish, you can target it with PCR. And if there's that fish DNA, it'll show up positive. If there's not that fish DNA, it won't. So it's super useful for that. You can do positive, negative, and things like that as well. Super cool. So DNA profiling is essentially using that PCR to look for regions in your DNA called STR, short tandem repeats. And essentially everybody has these. They're tiny little sections of DNA that are amplified for some reason inside your chromosome. They don't really seem to matter too much. People have different numbers of these STRs, these short tandem repeats. So they just compare the number of STRs that people have from crime scenes and things like that as well as their DNA sequence to help figure out paternity, to help figure out who committed a crime and things like that. So DNA profiling helps strengthen the criminal justice system. It helps convict people that are actually committed crimes, that they leave behind evidence. And more importantly, it helps exonerate people that are wrongfully convicted. People that have been sentenced to jail for murder and rape and things like that. If there was DNA evidence left behind in the 80s and 70s and 90s before people started really testing it, that was collected. And nowadays that they've started to test the DNA of those old cases, they find out that these people really aren't guilty and didn't actually commit the crime. They weren't there. It was somebody else's DNA at the crime scene and things like that. So they're released from prisons, which is an awesome thing to occur. Stem cells are super cool. Another way to deal with modern day problems. Lots of concern over stem cells. Some say they're good, some say they're not. Just depends on which side of the political spectrum you tend to fall on. Stem cells and cloning is super cool. You can make new copies of cells and new copies of organisms. And once again, really just depends on which side of the political spectrum you fall on that one. So what is a stem cell? Well, stem cells are essentially tiny little cells that are the very first cells to develop. When an egg is fertilized by a sperm, that little fertilized egg is going to start to divide very quickly. And the first couple of cells that are divided are going to be embryonic stem cells. They don't have a function yet. They are going to be tiny little baby cells that aren't programmed to become something yet. Eventually what will happen is this cell will divide, the embryo will divide and grow and grow and grow. And these cells over here will eventually develop into the brain, the neurons. This little blob right here might become the leg muscles, and this over here might trigger it to become a red blood cell. So they all turn into different cells eventually as the body grows. But prior to that stage, they don't have a function yet. And we as scientists have figured out how to take these particular undifferentiated cells and turn them into something else. We can force them to turn into things that we want them to do. So we can take these stem cells, these embryonic stem cells, and someone needs to grow a new liver. We can grow a new liver for them in a lab. We can grow a new heart in a lab, in theory. We can grow new body parts from these stem cells. We can allow them to replace damaged immune systems, which is what they do now. You've probably heard bone marrow transplants. It's essentially a stem cell transplant. They use your own stem cells, though. There's different versions of stem cells. You have your own stem cells. There's embryonic stem cells. Adult stem cells are a little less useful, but they're still kind of useful. They're not quite as good as these, but they work fairly well to replace the type of cell that you're targeting. So they work for bone marrow. But super cool stem cells. If we could figure out how to use these, we might be able to solve tons of diseases before they become problems. So we can go in and correct the problem in a developing embryo and insert the correct stem cell or fix the gene or things like that, or grow somebody back a liver on a transplantable liver in a lab. So super cool. Like I mentioned, there are also adult stem cells that are found in bone marrow. And you can turn these into closely related type of cells. So bone marrow is this type of stem cell. You can get bone out of it, fat cells, white and red blood cells, immune cells out of it, but you can't make really a heart out of those type of cells. So they're not quite as useful. But they're still useful in the sense of it's nice to be able to regrow your entire immune system. Or regrow someone else's immune system, but they're not as useful because you can't regrow liver and things like that from them. And now cloning is the process of creating an identical copy of an organism in a lab. Now, lots and lots and lots of plants and bacteria and little tiny microscopic organisms and things naturally clone themselves. Asexual reproduction is natural cloning. It's just literally making a genetically identical copy of yourself. Now, cloning, I tend to refer to that a little more as the laboratory term is of asexual reproduction. But cloning, in the sense of a lab, is really easy to do with plants. You can essentially just take a cutting off of a plant and stick it, it's like cut off a leaf or cut off a stem, stick it right next door in this little petri dish here, this agar dish, and it'll grow and make another plant. It's genetically identical to the original plant. Super, super easy to clone plants. If you garden, you know what I'm talking about with plant cuttings, plant clippings. Super easy to do this with plants. Animals, a little different. Essentially what you do is you take an egg from a mother. So let's talk about Dolly the sheep. You take an egg from Dolly the sheep, you remove the nucleus. All the DNA of Dolly the sheep, of the original mother sheep's DNA, is gone. So you have an egg with nothing inside of it, just an empty egg with no DNA inside. And then what they did was, so we're talking about Dolly the sheep that we're cloning here. They took a cell from the original Dolly the sheep, the one that's going to be cloned. And they took her cell from her arm, and they took the DNA out of it, the entire nucleus out of that cell. And what they did then is they stuck the nucleus from that cell into the empty egg from the original donor sheep. So now they have an egg with a nucleus inside of it from a different sheep. And then they sparked that nucleus or that embryo to life with an electric current. And now that embryo thinks it's been fertilized and has two sets of DNA inside of it now, a diploid cell instead of a haploid cell. So we took a haploid embryo and put a diploid nucleus inside of it so it thinks it's been fertilized. You spark it with an electric current, and then it starts to divide. It divides like it should. It starts to grow another cell. It's pretty neat. You stick it inside of the uterus of another sheep. Eventually it grows and develops, and a baby sheep will be born that is a genetically identical clone of Dolly, the original sheep that the cell was harvested from to start with. So super cool how this works. So check out the supplemental video on cloning. Human cloning could be cool, could not be cool. Depends, once again, on how you fall on the political spectrum. Really just depends on if you deem clones living organisms, or if they have their own autonomy, or what you like. Once again, how you feel about that one, where you fall on the political spectrum. DNA probes, so you can run your DNA in a lab, or you can run the DNA of a patient or an infant or something like that to figure out if they have any particular diseases that might be found inside their genome. So you can run these things with DNA probes. So essentially what you're looking for is you have a little segment that's tagged with a fluorescent label that looks for genes that are damaged. So you find a complementary section of the gene that's inside of here. So essentially what happened, let me kind of start over a little bit. You have your DNA chain here, A, T, G's, and C's. If you have a normal sequence of gene, you can see down here, A, T, G's, and C's, it's not going to line up with these little probes. If you have a damaged section of the gene, say you have cystic fibrosis, the probe will stick to it. And when the probe sticks to it, you will see a glowing part of the chromosome, which allows you to know that this chromosome is damaged because the probe stuck to it. If the probe doesn't stick, you know this person does not have a damaged allele, and not have a damaged DNA, and not have a damaged gene, because the probe did not bind. And you can see that here, non-damaged cell. This one has a damaged gene right there. So you can test for lots of different diseases as an embryo, as an adult, child, cystic fibrosis, the gene, hemophilia, Tay-Sachs, sickle cell disease. Lots of diseases are tested for looking for this DNA probe method. PGD, pre-implantation genetic diagnosis. Essentially what they're going to do, this is looking for problems with eggs and sperm before they unite. So they harvest eggs from a female and scan it for problems, look for the DNA and see if there's any problems inside of that egg. The same for a male. The sperm is harvested. They scan it for any genetic problems in a lab. If it's cleared and there's no genetic problems, they will unite the egg and sperm in a laboratory and then reimplant it back inside of the mother later on. So super neat. Gene therapy is going to be hopefully a very novel treatment in the future at some point in time. And this is using those modified viruses to correct problems. So essentially what we do is we take a virus that's been modified to contain a healthy version of a gene that's broken. So you can see this person here has cystic fibrosis. This is a lung disease. They have a gene that's broken inside of their lungs for producing too much mucus. So what we do as scientists is we take a virus, we modify this virus attacks lung cells. So we take this virus and we modify it. We take out the dangerous part. We stick inside a healthy version of the cystic fibrosis gene, not a broken one that causes cystic fibrosis. We stick back in the right one. And then we infect this patient willingly with this virus. So then this patient goes in, they get infected with this virus. The virus will then travel to their lungs, cause disease, quote unquote, but instead of making you sick because the dangerous part's been removed, all it's going to do is insert the correct version of that gene into your DNA strains inside of your cells. And then the cells are going to take that DNA strain and use it to do what they're supposed to do with it, to fix the broken part within themselves. So we, in theory, could fix and cure diseases like this in the future, or in some ways they can do this now. In the future, hopefully, we'll be able to do this a lot more and a lot more regularly, often, and a lot more successfully. CRISPR is a super cool way that people have figured out nowadays to modify DNA. This is a naturally occurring enzyme, CRISPR. What it does is it finds a particular segment. We can literally go down to the nucleotide level and go in and remove one particular A and change it to a T if we want to. And you can change and essentially write and target whatever you want to do inside of a DNA strain. If you want to change this particular DNA strain to read this or to have this particular gene sequence or whatever, you can do that with CRISPR. So in theory, we could essentially do this to make designer babies. We could allow our babies to engineer our babies to have blue eyes and purple hair and green skin if we wanted, with this technology. Now that's kind of unreasonable, but you get the idea. So it's very cool. We could also use it in theory to trim out damaged parts of genes and damaged parts of our DNA. Not to that level yet. It causes some side effects and things like that when it's used. It's not that precise yet, but in the future it might be, which is super cool. So there are lots of issues that are surrounding the ethical use of DNA technology. Really good to reduce human suffering, make us have healthier children, detect diseases early, hopefully cure diseases that don't have cures at the moment. But there are lots of issues that surround the stem cell research, the abortion debates that surround that one, playing God, all kinds of debates that surround this one. So that's pretty much all I've got for this PowerPoint. If you guys have any questions at all, feel free to send me an email and let me know, and I'll be happy to elaborate. If not, have a great rest of your day.